Okay, let's talk about neurodevelopment. Neurodevelopment is an ongoing process, and you'll probably hear this term plasticity. You'll hear this a lot as you explore the neurosciences. And it actually means that the brain can modify itself. It can modify its connections and it can rewire itself. Okay, it is a complex process, and experience does play a key role, right? Um, and then lastly, there are dire consequences when something does go wrong. So we're going to talk about neurodevelopment. We're going to talk about the 5 phases that you see on the slide there: induction of the neural plate, proliferation, migration, axon growth, and then neuron death and apoptosis. Okay, but we will end this lecture also with a couple of neurodevelopmental disorders. Okay, let's start off with the induction of the neural plate and neural proliferation. So a patch of tissue on the dorsal surface of the embryo becomes the neural plate. You can see this in the graphic on the right-hand side. Visible 3 weeks after conception, you see this neural plate. And then neural plate cells are often referred to as embryonic stem cells. You've probably heard that term before, because they can become any kind of mature cell. You've probably heard of the fetal transplants where they're using these embryonic stem cells. The neural plate folds to form the neural groove, which then fuses to form the neural tube. Okay, and you see that again on the graphic. Inside will be the cerebral ventricles. Okay, and then the neural tube proliferates, and 3 swellings occur at the anterior end, which become the forebrain, midbrain, and hindbrain. Okay. So once cells have been created through cell division in the ventricular zone of the neural tube, they migrate. So now we're focusing on neurodevelopment, migration, and aggregation. So migrating cells are immature. They lack axons and dendrites, and they migrate long distances. After migration, cells align themselves with other cells and form structures. Through the use of cell adhesion molecules, or CAMs, this is how migration and aggregation occur. Okay? And then gap junctions are also used and are very prevalent in brain development. And these are the junctions where the 2 neurons are actually touching one another. You don't have the synapse in between. Okay, so we do know that aggregation is aided by these gap junctions. Okay. Then once migration is complete and structures have formed, the aggregation part, axons and dendrites begin to grow. So now we're on to the portion or the phase of axon growth and synapse formation. The growth of cone, or the growth cone, sorry, at the tip of each extension extends and retracts, trying to find its way. Again, the picture at the right shows you those growth cones. Chemoaffinity hypothesis states that postsynaptic targets release a chemical that may guide axonal growth. Formation of new synapses depends on the presence of glial cells, especially astrocytes. Remember, those were the large star-shaped glial cells, which supply the cholesterol. Okay. Now we talked about axon growth and the synapses and so on. Now we're looking at neuron death and synapse rearrangement. Okay, so approximately 50% more neurons than are needed are actually produced, and therefore a lot of neurons die off. Neurons die through a process called apoptosis, okay, which is a programmed cell death due to failure to compete for chemicals provided by targets. So the more targets, the fewer cell deaths. And destroying some cells increases the survival rate of remaining cells. Okay. So if you look at the figure on the right-hand side, you'll see a green, a yellow, and a blue neuron at the top. And you see how their axons feed down to the purple neurons there. And let's say we take, for instance, just the blue one. You'll notice the blue one is actually affecting the purple, the fourth purple neuron, the one to the furthest to the right. And then we could say it's also affecting the third one, but it's also going over and having an effect on the second and the first one. Okay, so it's very, very diffuse where it's making these connections. Okay, then if we look to the right, you'll notice how now you have still the green, the yellow, and the blue neurons, but now that blue neuron is only affecting the third and fourth neuron. Okay, so it's kind of like a pruning process here, which adds specificity. So neurons that fail to establish correct connections are particularly likely to die. But remember, there's 50% more neurons than are actually needed. Okay, space left after apoptosis, or this programmed cell death, is filled by sprouting axon terminals of surviving neurons. And that's what we see also on the in the figure. This ultimately leads to increased selectivity of transmission. Okay, now here I mentioned to you neuroplasticity, and that you would hear this term quite often. It means that the brain is plastic or changeable. Okay, examples of plasticity: adult musicians who play instruments fingered by the left hand have an enlarged representation of that hand in the right somatosensory cortex. So the somatosensory cortex actually changes and molds. Okay, remember it's plastic because of some extremity that's being used a lot more often. Okay, and in the case of a musician, let's say playing a guitar or a violin, is fingering all those strings and causing a lot more activity in that somatosensory cortex. Skill training leads to reorganization of the motor cortex as well. Okay? And that makes sense because the motor cortex is telling those muscles what to do. Okay? So neurogenesis, or growth of new neurons, is seen in the olfactory bulbs and the hippocampus. So not only is the brain plastic, it can change, but we now know that new neurons can grow. Okay? We don't see it all over in the brain, primarily olfactory bulbs, primarily hippocampus, but 10, 15 years ago, we didn't think that this would be possible at all. Okay? Enriched environments and exercise can also promote neurogenesis. So the figure on the left-hand side is just showing you how in green we have glial cells, in the blue we have existing neurons, and then you see the red neurons that are stained, and those are the new ones. Those are brand new neurons that are just growing. Okay. And then, so now we switch gears a little bit and we talk about disorders of neurodevelopment. Like we said, there's the 5 phases, okay? And then on one of those points of interest at the very beginning, we talked about when things go wrong, sometimes they can really, really go wrong, okay? So neurodevelopment is very specific and can be altered by certain things. So let's look at one of those neurodevelopmental disorders, which is autism. Okay, the incidence here is 6.6 per 1,000 births. 80% of those affected are males, 60% are mentally retarded, 35% are epileptic, and 25% have little or no language ability. Now, you've probably heard of autism as autism spectrum disorder. It is on a spectrum. It's on like a continuum. You've probably also heard the term Asperger's syndrome. Okay, and the reason why I put this on there is because you do hear this term so often, and it is a mild form, okay, of autism in which cognitive and linguistic functions are well preserved. Now, the 3 overarching core symptoms of autism are reduced ability to interpret emotions and intentions, reduced capacity for social interaction, and preoccupation with a single subject or activity. Okay. Now, applied behavioral analysis is something that is very, very popular. Many companies using this now, where it's an intensive type of behavioral therapy, and it is showing some promise, okay, for helping individuals with autism. Most of these individuals have some abilities that are preserved. For instance, like rote memory, the ability to do jigsaw puzzles, musical ability, artistic ability, and in fact, some of them have preserved abilities that are far greater than any other normal person, okay? And we would look at these people, or we term this autistic savants, okay? And are intellectually handicapped, okay? They still have some of these deficits, but they also display a specific cognitive or artistic ability, okay? And approximately 1 in 10 autistic individuals display savant abilities. So here it could be they might be able to look at a scene and then just memorize that. Maybe their spatial ability is fantastic. Maybe the ability to have perfect tone or pitch, music ability, their ability to model. So they listen to a song or they listen to somebody play a piano and they can play it right back. Okay, that would definitely be an example of an autistic savant. Okay. Some genetics and neurological mechanisms here for autism. Siblings of the autistic have a 5% chance of being autistic, okay? But there is a 60% concordance rate, okay? So identical twins, if one is autistic, 60% chance that the other one is also autistic. Several genes in interaction with the environment have been viewed. This is not a single gene issue, and the etiology for autism is still really not known, but we do know that it is neurodevelopmental, okay? Because we have looked at some of the brain structures here. The cerebellum is involved. That makes sense for some of the odd motor movements, like the clapping of hands and so on that we see. The amygdala, that has to do with the limbic system and emotion, and we know that's a factor. And then the frontal cortex is for higher order and critical thinking, and there might be, in this case, a decreased amount of activity in the frontal cortex. There are 2 lines of research on cortical involvement in autism. There's the abnormal response to faces, so they spend less time than non-autistic children or subjects looking at faces, especially the eyes, and then low functional MRI activity in the fusiform face area. And then second, possibly deficient in mirror neuron function. So this is these are the neurons that fire when you are observing somebody else doing an activity. Okay, and then the last neurodevelopmental disorder that we talk about is Williams syndrome. Okay, this occurs 1 in every 7,500 births. Mental retardation and an uneven pattern of abilities and disabilities are observed. People with Williams syndrome are sociable, empathetic, and talkative. They exhibit language skills, music skills, and an enhanced ability to recognize faces. However, profound impairments occur in spatial cognition, the ability to understand, reason, remember spatial relations among objects or space. If you look at the figure on the right-hand side, you'll see the areas in red are reduced cortical volume. So we see a general thinning of cortex at juncture of occipital and parietal lobes and at the orbital frontal cortex. Also, another key characteristic of this is their appearance, oftentimes short, small, oval ears, broad mouths. Okay, so those gives you just an example of neurodevelopment and how that process is a very critical process, and sometimes it does go wrong and we see these disorders.